So hello students, all of you, welcome to your own channel, right? Vedantu Science Olympiad. This is Basavaraj sir, your biology master teacher. Now, students, all of you have ever wondered, we always say, right? People always say, eat food, it'll give you energy. Everyone says, if you eat food, it gives you energy. But how? How does the food gives you energy? See, all of you should be know that in our body, there is something called as ATP molecule, adenosine triphosphate molecules. These molecules have energy. But from food, students, from food, how do we get this energy? Right? So this is what we'll be learning in today's class. In today's class, I'll be teaching you how do we obtain energy, or how do plants and animals obtain energy from your entire food items. Okay, students. Now, before we start, students, I want all of you to understand the basic difference between breathing and respiration. First of all, breathing and respiration are not same to same. Breathing and respiration are two separate phenomena. For example, students, for example, I want all of you to breathe in. Now, this is called as breathing. Breathing is a much more physical process, right? Breathing right now is a physical process. It is a physical process. Now, what about respiration, students? Respiration is a chemical process. It is a chemical process. Students, today's class, I'll teach you what is the respiration actually. I'll teach you what is exactly is respiration today's class. Okay. Now, what is the purpose of breathing? The purpose of breathing is to take in oxygen, remove out CO2. Yes or no, students? To take in oxygen, to remove out CO2. Yes or no? Now, what is the purpose of respiration? The purpose of respiration is to produce energy. To produce energy. Right? To produce energy is the purpose of respiration. Now, you might ask, sir, where do you think breathing happens? Breathing has multiple parts, right? Nostrils, oxygen goes in, lungs, which take part in your mainly breathing. But respiration, students, where do you think respiration happens? Respiration is a process which happens inside a cell. Students, respiration is a process which happens inside a cell. All of you students, all of you. Respiration, students, right? Which book is the best for students? Recently, one of the videos that we discovered, students. Yes. Olympiad is slaying everywhere. Yes, students. Tatva is really good. Students. All of you should remember, respiration happens inside a cell. All of you should know, respiration is a process which happens inside a cell. Please remember that, students. Please remember that. It happens inside a cell. While breathing happens is a physical process. That is breathing. Okay, students. Control, students. Can we control breathing? Yes, we can control breathing. Like, we can hold the breath. We can take in the breath. Yes, we can control this. But can we control respiration? No. Respiration cannot be controlled at all. It is happening. Kanya, hi. Kanya, hi. Respiration cannot be controlled at all. Respiration is a process which keeps on happening. You cannot control at all, students. You cannot control at all. Okay, you cannot control at all respiration. Okay, yes, students. Does breathing gives you energy? No. Does respiration gives you energy? Yes, respiration gives you energy, students. So I believe all of you know the basic difference between breathing and respiration right now. Breathing is your physical, respiration is chemical. Okay, respiration is a chemical process where energy is produced to all of you. Okay, let me tell you a small thing. How does it actually happens? So Tattva contains all this theory. Yes, it does. Okay. Now, so students, for example, what exactly is respiration? If you ask me a definition of respiration, students, the definition of respiration is it is a biological oxidation of organic matter. Biological oxidation of organic matter is the food. Very complex definition. I'll make it easy for you. Understand. Let's draw one cell right now. Let's draw a cell. This is a one cell, student. This is a one cell right now. Students, tell me, what did you eat for lunch? All of you. What did you eat for lunch? Students, what did you eat for lunch? Students, quickly smash the like button also. Quickly smash the like button right now. Quickly smash the like button. Okay. Now let's say for lunch you ate rajma chawal. Okay. You ate rajma chawal for your lunch. The food is entering inside a cell. Similar students, similarly, even oxygen enters inside a cell. So oxygen is also entering inside a cell. Food is also entering inside a cell. Now this oxygen will act on the food. The oxygen which is entering inside a cell. This right here is a cell. All of you know what is a cell. Now once this food acts on the once the oxygen acts on the food undergoes something called as oxidation. Students, if you do not know what is oxidation, I'll teach you today. I will teach you what is oxidation today. Okay. Now it undergoes oxidation. When the food is oxidized completely, this food will release energy. The food will release energy. And this energy, students, this energy, you cannot take it as such. You cannot eat the energy. No. This entire energy is stored in the form of ATP, adenosine triphosphate. Okay. So, food is entering inside a cell. Oxygen is entering inside a cell. There is oxidation. That is basically carbon-carbon bond is breaking here. That is, CC bond is broken. It is broken here. The carbon-carbon bond is broken here in oxidation, and it gives you energy, and this entire energy is stored in the form of ATP. The energy is taken, it is stored in the form of ATP. Okay, that is what happens inside a cell. Now students, in this entire oxidation, I say, right, oxidation, there are multiple steps. Let me just tell you once and for all. There is glycolysis, there is a link reaction, there is Krebs cycle, there is entire something called as electron transport chain. But all of that will not be teaching today. Today's class, only one concept today. The only concept today is called as glycolysis. So glycolysis is a process which comes under your oxidation right now here. Okay, that is your glycolysis, students. Okay, students, I want all of you to know this is an ATP molecule. This is how ATP molecule looks like. ATP molecule. This is the ATP molecule, adenosine triphosphate. Here in this tail, all the energy is stored in this tail. All the energy we say, energy for our body, it is present here only, students. Okay. Now, students, I want all of you to know. Do all of you know there is something called as a check? Students, do all of you know what is a check? Check. Have you seen check in your life? Check. If you take the check and you go to bank and you give the check, what will happen, students? All of you tell me in the chat. You take a check, you go to the bank and you give the check. Do they give you money? Yes, they give you money. Okay, they will give you money. Yes or no? They'll give you money, right? So I want all of you to understand what is the difference between ATP and NADH or FADH. Okay. Now, students, I want all of you to understand. Inside our body, there is a currency. Like every single organ, if they have to do something, like students, you tell me in the chat right now, if all of you have to go buy ice cream, go buy chips, to go buy books, do you need money? Yes, you need money. Similarly, inside our body also, every single function requires money here. Every single function, excretion, your neural connections, everything requires energy. And the energy or the currency, they also require money. And the money here in a body is called as ATP. So ATP is also called as energy currency of the cell. Like for example, inside of a cell, energy currency of a body, any process inside our body, if it has to happen, it needs currency, and the currency is called as ATP. Okay, and this ATP has a lot of energy. So our body will use the ATP and it will do the work. Like how you will use the money and buy the ice cream, you will buy the samosa, yes or no? So you will use the money and buy things. Our body will use ATP and do things. Okay, and students, now what is the difference between ATP and NADH and FADH, students? This NADH and FADH is like a check. NADH and FADH is like a check. Okay. Now this NADH and FADH is a nicotinamide adenine dinucleotide. This NADH is there, right? If you take the NADH molecule and you give it to electron transport chain, if you take NADH and give it to electron transport chain, students, it will give you ATP. So let's how, if I take this check and I go to bank and I deposit the check, will I get the money back? Yes. So if I use NADH, I will give ATP. So from NADH I can get ATP. Like from check you can get cash. From check you can get a cash. From NADH you can get ATP. Now from one NADH you can get 3 ATP. You can get 3 ATP. And from students, from one FADH to you can get 2 ATP. Okay. So students, just try NADH in Google, you'll get it, friends. Nicotinamide, nicotine, NICOTINE, nicotinamide, AMIDE, dinucleotide, DI, nucleotide, NUCLEOTIDE, right? Nicotinamide adenine dinucleotide. Okay. So NADH and FADH2, right? One NADH will give you 3 ATP, one FADH will give you 2 ATP. From check you get cash, from NADH you get money. Okay. Now students, now we come to the first step in all the respiration. It could be aerobic respiration, it could be anaerobic respiration, right? Every single type of respiration, every single organism in the world, bacteria, prokaryote, eukaryote, unicellular, multicellular, plants, animals, this process will happen in all the organisms, no exception. Okay, this process will happen because every single organism, if the organism has to survive, it needs energy, and this is how they can get energy. See, there are students, there are multiple steps actually. There are multiple steps. There's a glycolysis, Krebs cycle, ETC, link reaction, all that is there. But this right here is the first step in respiration. The first step in all of respiration is called as glycolysis. Okay, students, it is called as glycolysis. Now, students, I'll teach you what exactly is glycolysis. Understand very carefully, students. Very carefully, all of you need to understand. Okay. Now, what exactly happens in the case of glycolysis, students? Now, the meaning of glyco means sugar. Lysis means to break. So the whole point of glycolysis is to completely break a sugar. Now students, if you have attended my previous class of biomolecules, can anyone give me an example of very simple sugar? Let's say glucose. So if our body is taking glucose, the glucose is broken down in this process. So glucose is entering. The glucose is actually broken down in glycolysis. Piece of glycolysis is happening. Piece of your glucose is happening, students. Okay. Now you might ask me, sir, where does it happen? What is the location, student? Location. It happens in the cytoplasm. It happens in the cytoplasm of cell. Cytoplasm of cell, students. Cytoplasm of cell. Now, how many steps are, students? Number of steps here. This reaction has how many steps? It has 10 steps. It has 10 different steps. Students, here it happens in the case of eukaryotes, prokaryotes, plants, animals, everywhere. In all the organisms, this happens, students. It happens in all the organisms. Okay. Prokaryotes, eukaryotes, plant, animals, it happens. Okay, students. Okay. Students, you didn't attend. So please attend my biomolecules chapter where I've taught you what does it mean by glucose, what do you mean by sucrose. Sucrose is a disaccharide. Sucrose is a disaccharide. I'm talking about monosaccharide. Monosaccharides are like your glucose, fructose are monosaccharides. Okay. So Tejas, please attend. Tejas, please attend my biomolecules chapter to understand all these topics also, because that is very important. Biomolecules is very important. It's a fundamental. Okay. Now students, this entire glycolysis is also called as EMP pathway. It is also called as EMP pathway because of 3 scientists who discovered this process. Okay. Gustav Emden, Otto Meyerhof, Jacob Parnas. So EMP, this first letter of this second name, EMP has been taken. It's called as EMP pathway. Okay. Now students, before I start this entire complex process of glycolysis, I want all of you to understand 2 different words here. What is the meaning of oxidation? What is the meaning of reduction? Okay. Now, oxidation basically means loss of electrons or loss of protons or gain of oxygen also. Gain of oxygen also same meaning is there. But for now we'll remember oxidation is loss of electrons and protons, right, in sometimes accompanied by loss of protons. Hydrogen is a proton, H plus is a proton. Okay, proton. Now what is reduction? Reduction, students. Okay. Now what is reduction? Reduction is the gain of electrons. So reduction means gain of electrons and gain of protons. So reduction means gain of electrons or gain of protons, or hydrogen here. Okay, that is reduction. Oxidation is loss. In oxidation you are losing. Students, is it clear to all of you? Oxidation is loss, reduction is gain. Simple difference to all of you. Now students, now the entire glycolysis can be divided into two stages. The first stage is called as your payoff. First stage is called as preparatory stage. The first stage is called as preparatory stage. The second stage is called as payoff stage. This second stage, first stage. Okay. Now students, you tell me, in your house, do they invest money in your house? Do they invest money? Yes or no? Like you take the money, you invest somewhere. After investing, you get a return back. So invest, return, invest, return. So here also, the first stage is called as preparatory stage. In preparatory stage, we invest. Investing is happening here. Now, once investment has been done, in payoff, you get the returns. You get the returns back in payoff stage. That is the basic concept of preparatory and payoff. Okay, preparatory and payoff. Students. Now we will understand this is the entire glycolysis. This is happening in your body right now. This is happening in your body right now, students. Okay. Your parents investing on you. That's amazing. They just students, see this. This entire process is happening in your body right now. Actually, right now it is happening. Okay. So be happy. You are learning something which is happening inside you right now. Okay, it's happening inside your cell right now. Inside your cell. Okay. Now in preparatory stage, there is step 1. In step 1, what happens, students? In step 1, glucose, which is a 1, 2, 3, 4, 5, 6 carbon molecule. Glucose is a 6 carbon molecule, gets converted to glucose 6 phosphate. How? Very simple, student. Very simple. Now this is a glucose here. Now I will take a ATP molecule, adenosine triphosphate, and I'll break this phosphate here. I'll break it. Now I will take this phosphate and I will attach it to the 6th position here. So glucose becomes glucose 6 phosphate. That is the first step, student. That is the first step. Now if I take ATP, right, I'll share the PDF. Don't worry, students. I'll share the PDF. If you take the ATP molecule and you remove one phosphate from adenosine triphosphate, will it become adenosine diphosphate now? Students, understand? See, where it is. If this is a ATP molecule, it is called as ATP. Adenosine, this one, sugar here. Tri means what, students? Tri means what? Tri means 3. Tri means 3. Phosphate, 1, 2, 3. So ATP has 3 phosphates in it. So ATP, if I remove this phosphate out, if I remove this, what will it become? ATP will become ADP, adenosine diphosphate. Di means what? 2 phosphates are here. 1, 2, 2 phosphate. So diphosphate. So ATP becomes ADP. ATP becomes ADP. That is what is happening here, students. This ATP, if I remove this phosphate and I take this phosphate and I attach it here, what happens, student? This is what happens. We get ADP here and we get glucose 6 phosphate. Okay. So glucose to glucose 6 phosphate is done. In this, there is an enzyme called as hexokinase. Is the enzyme here. Hexokinase is the enzyme. Enzyme is a like catalyst. Catalyst in chemistry, you will write chemistry, you learn catalyst, which will increase or decrease the rate of reaction. Same thing. Here we have hexokinase here. Hexokinase. Okay. So that is the first step. Glucose to glucose 6 phosphate. First step is done. Now, students. Now, what is second step, students? Second step is called as isomerization. Now, what do I mean by isomerization? Understand the meaning of isomer. Okay. The meaning of isomer, students. The meaning of isomer, students. Okay. Here, isomer is nothing but isomers are molecules with the same chemical formula. This and this, both of them have same chemical formula. It is C6H11O9 phosphate, right? Both of them, glucose 6 phosphate and fructose 6 phosphate, have the same chemical formula, right? They have the same chemical formula, but what is the but here? But the arrangement is different. See, but different structural formula. Those are called as isomers. Those are called as isomers, right? Isomers are molecules with same chemical formula. Both have C6H11O9P. Same chemical formula, but arrange, can you see here we have a ketone group, here we have a aldehyde group, here aldehyde. See, the arrangement is different, but chemically same. That is called as an isomer. So glucose 6 phosphate gets isomerized to fructose 6 phosphate. That is step 2 here. Can you see? Glucose to fructose. Glucose to fructose. Okay. And friends, remember, every single time you do isomerization, there is never energy exchange. Never energy exchange. Can you see? Glucose to glucose 6 phosphate, glucose 6 phosphate to fructose 6 phosphate, there is no change in energy. Okay. Now students, now this is fructose 6 phosphate here. Okay. Now to this fructose 6 phosphate, I will take one more ATP and I will break this again. I'll break this. I'll take this phosphate and I will add it to the first position. See students, this is called as glucose, fructose 6 phosphate. If I take this phosphate and if I add it here, what will become? It will become fructose 1,6 bisphosphate. It becomes bisphosphate, right? It can be changed. It can be changed. It can be in chain also. Okay. So fructose 6 phosphate becomes fructose 1,6 now, because this phosphate is attaching to the first position. So fructose 1,6 bisphosphate it becomes. Okay, it becomes bisphosphate. Okay. That's why it's sufficient actually. Okay, it becomes 1,6 bisphosphate. 1,6 bisphosphate. So that is the quick summary till now. Quick summary till now is glucose 1 ATP is given here. We got glucose 6 phosphate. Glucose 6 phosphate undergone isomerization to fructose 6 phosphate. To take this one more ATP and we add one more phosphate here, it became fructose 1,6 bisphosphate. It became fructose 1,6 bisphosphate right now. 1,6 bisphosphate. Now till here, this is a story right now. Till here, this is a story right now. Now students, now can you see here? Glucose 1 ATP consumed here, minus 1 ATP. Here, minus 1 ATP here also to make this fructose. And right now we have something we have right now at fructose 1,6 bisphosphate. This is where we are right now. We are at this point right now, students. Okay, we are at this point right now here. Fructose 1,6 bisphosphate. We are here right now, students. We are here right now. Fructose 1,6 bisphosphate. Okay. Now, students, what happens? Understand, students. Now, this entire fructose, this is how it looks. Carbon, 3, 4, 5, right? 6. Here we have a phosphate. Here I have a phosphate. Now, students, what is happening is this 6 carbon molecule, fructose 1,6 bisphosphate, it is actually breaking into half. It is actually breaking into half. This 3 on the top, this 3 at the bottom. So this entire thing is splitting open right now. It's split open. Can you see? Split open right now. This 3 on top, this 3 at the bottom. That is what happening here right now, students. So fructose 1,6 bisphosphate is splitting into 2 parts. The first part is called as students, right? The first part is called as your glyceraldehyde 3 phosphate. The second 3 carbon is called as dihydroxyacetone phosphate. Students, you have to remember all this. You have to remember all this. Questions might come from here. Questions might come from here, students. You never know. Okay, so you have to remember this. Okay, so fructose 1,6 bisphosphate becomes, this is called as your GAP, glyceraldehyde 3 phosphate. This is called as DHAP, dihydroxyacetone phosphate, DHAP, 2 molecules. Now, students, the funny part is these 2 are also isomers. They can interchange among each other. They interchange among each other. Yes, they can. So for now, convenience sake, I will convert DHAP into GAP. I will convert DHAP into GAP, that is dihydroxyacetone phosphate. Dihydroxyacetone phosphate, I will convert it to GAP right now. Okay, I will convert it to GAP, students. Okay, they are isomers. So we can interchange, we can convert this to this also. There is no issue. We can convert this to this also. There is no issue. But for now, I will convert DHAP into GAP. So from now onwards, I will always write into 2 from here. Why into 2? Because right now I have 2 molecules of GAP here. I have 2 molecules of GAP. How come 2 molecules, students? Because this DHAP is converted. This is 1 molecule. If I'm converting this to this here again, if I'm converting, students, if I'm converting DHAP into GAP here, so can I tell 1 molecule, this 1 molecule? So in total we have 2 molecules of GAP from here. We have 2 molecules of GAP right now. Can you see here? From this DHAP will now get converted to GAP. Okay, it's getting converted to GAP. So for convenience sake right now, fructose 1,6 bisphosphate, 6 carbon molecule. This 6 carbon molecule is converted to 3 carbon molecule. This is 3 carbon molecule. This is 6 carbon molecule. 6 carbon molecule is divided into 3 carbon 2 times. 3 2s are 6 here. Okay, 6 carbon. Now students, what happens? Now the payoff starts. Now you will start getting the money back. All the investment you did, right? Investment. Now you will get the money back again to you. Now what is the first step you're getting money? Now students, what happens? This GAP is there, right? GAP. It undergoes oxidation. Now students, can anyone remind me what is oxidation? Oxidation is loss of electrons, loss of protons. Yes or no? Say students. This entire GAP, glyceraldehyde 3-phosphate, 2 molecules of it, 2 molecules. GAP undergoes oxidation right now. Oxidation. Now once it undergoes oxidation, electrons are released, protons are released, energy is released. GAP is undergoing oxidation. Now students, I want all of you to understand what will happen to this electrons, protons, energy. What will happen to them? Understand, students, understand. These electrons and protons are now taken up by electron carrier called as NAD+. They are taken up by NAD+. This is, remember students, I told you the NADH, same thing. This is the electron carrier part. This is the oxidized state one. Okay, nicotinamide adenine dinucleotide. This is NAD+. This NAD+ will take these electrons, will take the protons. Okay. Now students, if this NAD+ is taking the electrons and protons, can I tell it is gaining them? This NAD+ is actually gaining them right now. Gain. Now, what do I call gain of electrons and gain of protons? Gain of electrons and gain of protons is called as what? It is called as reduction. Reduction. Yes or no? So it will get converted. NAD+ will take the electrons. It will get converted to NADH. It will converted to NADH, students. So let me just recap on what, students. Then that is the fate of these two. Now what about the energy, students? Now this energy is used, students. This energy is used right now. This energy is used to bring one inorganic phosphate. Inorganic phosphate from the cytoplasm. Inorganic phosphate from cytoplasm, and I will take this phosphate and I will add it to the first position here. I will add it to the first position. Okay, oxidation, and we end up getting something like this. So from glycerol, 2 molecules of glyceraldehyde 3-phosphate, I will get 2 molecules of 1,3 bisphosphoglyceric acid. 1,3 bisphosphoglyceric acid. Students, again, if you're learning this for the first time, all of these words are difficult to understand, I know. But as the time goes by, as you read it a couple of times, you learn the glycolysis a couple of times, it becomes easy for you in the future. But right now, it may be difficult, I know. Okay, first time if you're learning. Okay, let me just show you one more time what happens, students. This 2 glyceraldehyde 3-phosphate here, glyceraldehyde 3-phosphate undergoes oxidation. Okay, oxidation is what, students? Oxidation is loss of electrons, loss of protons. Electrons, protons are lost from here. Lost. Now these electrons and protons are taken up by NAD+, which undergoes reduction now, because it is now gaining these electrons and protons. These electron protons are released from where? They are released from here, gap. Okay, they are taken up by. Now this undergoes reduction to form NADH. Okay, this energy is used to bring inorganic phosphate from the cytoplasm and attach it here. And students, we end up getting from 2 glyceraldehyde 3 phosphate to 1,3 bis phosphoglyceric acid. Okay, so all of you can see, students, here 1,3 bis phosphoglyceric acid. Now this is for one molecule, but we are doing it 2 times because this is 2 molecules. So we end up getting 2 NADH, not 1. So from one molecule you get 1 NADH. From one glyceraldehyde, from one gap you get 1 NADH. Since we have 2 gap here, so 2 NADH we get here, not 1, 2 NADH. Okay, that's what it is now. Now what happens, students? This 1,3 bis phosphoglyceric acid is there, right? 1,3 bis phosphoglyceric acid. This bond is unstable, so this bond will break. Once this bond breaks, right, this phosphate is now given to a ADP. Students, all of you tell me in the chat what will happen now. If this ADP, this phosphate joins here, what do we get? From adenosine diphosphate, it will become ATP again, ATP. Okay, so ADP to ATP conversion happens here. Okay, so you can see here also, students, you can see here from 1,3 bis phosphoglyceric acid to, if this is broken, what do we have? We only have 3 part here. We only have 3. So we end up getting what, students? We end up getting 3 phosphoglyceric acid. So from 1,3 we get 3 because 1 is removed now. This is removed right now, right? This is removed. It is removed here. Okay. So from 1,3 bis phosphoglyceric acid we get 3 phosphoglyceric acid because one phosphate is removed from there. So we get 3 phosphoglyceric acid and 1 ATP is given here. Now, student, this 1 ATP is for 1 molecule transfer. But do we have 1 molecule? No, we have 2 molecules. So into 2 again ATP also, into 2 NADH here also. Okay. Now students, what happens? Next step is again isomerization. Okay. Now 3 phosphoglyceric acid, this 3 is transferred top. So 3 will become 2 phosphoglycerate. So from 3 phosphoglyceric acid to 2 phosphoglycerate becomes again isomerization, no energy transfer. Okay. Now students, from this 2 phosphoglycerate we remove water molecule. It becomes PEP. PEP is phosphoenol pyruvate. Now from this phosphoenol pyruvate, again students, this phosphate is again broken out. It is broken out. Now once this phosphate is broken out, students, what do we get? See here, it is broken out. So phosphoenol pyruvate, PEP, now gets converted to pyruvate because this P is broken here and it is given to this here ADP. So we get ATP again in the last step also. So in the last step also we get pyruvate in the end or pyruvic acid and again 2 ATPs here. Why 2? Because 2 molecules everywhere from here onwards. So that is your entire glycolysis, starting from glucose ending in pyruvate. That is the entire story of glycolysis. Starting from glucose ending in pyruvate. Now that is the entire glycolysis. Again students, if you're learning for the first time, it will be difficult. So please go through the video one more time, download the notes, right? Try understanding glycolysis even deeper. Okay, students, here this is your entire glycolysis. I given one more time here. Glucose to glucose 6-phosphate, fructose 6-phosphate, isomerization here, students. Fructose 1,6 bisphosphate here. Then what happened? When we get glyceraldehyde 3-phosphate, 1,3 glyceraldehyde 3-phosphate, 3 phosphoglyceric acid, 2 phosphoglyceric acid, phosphoenol pyruvate and pyruvic acid. Preparatory, payoff students. Here we get fructose gets converted, students. Remember, fructose gets converted to 2 molecules here, DHAP and GAP. It happens here only. DHAP and GAP happens here only. We can see here DHAP. Okay. Now, sometimes in examination they can ask you what is the net profit here. Okay, let's see. What is the net profit, students? What exactly is the net profit, students? Understand. In preparatory stage, -2 ATP. Yes or no? Here, you can see. Here one ATP is lost, here one ATP is lost. In two places the ATP is lost here. So minus 2 ATP, students. Minus 2 ATP. Now in payoff, we get 2 NADH. We get 2 NADH here, 2 ATP here, 2 ATP. So in total we get 4 ATPs we get 4 ATPs we get in total here. And I told you students, 1 NADH will give you 3 ATP. 1 NADH will give you 3 ATP. So 2 NADH will give you how much? 3 to the 6 ATP. This will give you 6 ATP. 1 NADH gives 3 ATP. So 2 NADH will give you 6 ATP. Okay. So students, if you add everything here, 6 plus 4 is 10 ATP. This is the gross. Now if you remove the minus 2 ATP from the preparatory, 10 minus 2 is 8 ATP. This is the net gain of energy. This is the net gain of energy from glycolysis in our body, in any body. Net gain of energy from glycolysis. Okay, students. Now, here again, students, what other questions they can ask you? They can ask you which step ATP is formed, ATP is consumed, 1 and 3. Which step ATP is produced, 7 and 10. Rate determining step is third step because it is the slowest step. Irreversible steps are 1, 3 and 10. We cannot change them back. Oxidation happens in the sixth step, that is PgAl2 1,3 bisphosphonacic acid. Oxidation happens there where we get NADH. EMP pathway because of scientist. Which step water is formed? 9th step. Aerobes and anaerobes, it happens in both of them. It happens. First enzyme is called as hexokinase. Last enzyme is called as pyruvate kinase. Students, after this, they will not ask you any other questions. All the questions from glycolysis is covered here itself. Okay, it is covered here, students. Again, students, I'm telling you, glycolysis is a very detailed depth concept here. So whatever you have learned right now is a very high level concept. It's very difficult to understand at one go. It'll take some time. Watch the video one more time for a better understanding, students. Okay, so thank you so much, students. I'll see you in the next class. Tomorrow we'll be doing biomolecules part 4, the final part tomorrow, where I'll be teaching you what exactly is the enzymes, what is the catalyst, how do they work, what is the activation energy in tomorrow's class. So until then, students, take care all of you, keep studying, and bye bye all of you. Bye bye.